Alright, hey everybody. Welcome to another video lecture. This is chapter 22. We are going to look at some metabolic pathways for carbohydrates. Alright, so let's just dive right in. I always do that, never fails. Okay, dive right in. So let's talk about some terms. Metabolism is both the breakdown and the buildup of our body's molecules. Catabolic metabolism is the breakdown. So catabolic reactions are going to be those that are breaking things down, and anabolic reactions are those that build things up. One of the key differences is that anabolic requires ATP, and generally catabolic is going to provide ATP. A few stages of catabolic reactions. There's the digestion, hydrolysis, or essential breakdown of big molecules to little ones to get into the bloodstream. Then once they can get into the bloodstream, they can get into the cells, and then they're broken down into smaller chunks. These are 2 and 3 carbon compounds, and then these get oxidized into carbon dioxide and breathed out in the citric acid cycle, which I don't believe is a subject of this chapter. It might be, we'll have to look. So I think this has glycolysis in it, but we'll see. And then electron transport. That is the objective of citric acid cycle, is to provide electron transporting molecules to the site of ATP synthesis in our mitochondria. So stage 1, stage 2, stage 3. So stage 1 is actually pretty easy, right? We're just going to look at some of that mechanical breakdown, right, that our teeth do, and then some of the acid digestion, and then some enzyme digestion. We're going to focus on carbohydrates. So obviously, different protein or different sources of energy would be your carbohydrates, your proteins, and your fats. These things would all be broken down in the same sort of way through mechanical breakdown in your mouth with your teeth, and then sort of that acidic environment of the stomach. Once those get into smaller bits, proteins, amino acids, polysaccharides, monosaccharides, and lipids into fatty acids, then they can go about being oxidized, so entering into our catabolic reaction cycles. And then once we can get essentially acetyl-CoA, this is the molecule that's going to be entering the citric acid cycle from all of our branches here, and even some other molecules that can enter in on the protein side can be broken down in the citric acid cycle for energy. So acetyl-CoA is ultimately what we're trying to get to. I think we're going to get to pyruvate. Can't spell pyruvate today. As far as the cell structures go, required for metabolism, we have a few. These are usually mitochondria in our bodies, but some of the similar features that they have is they must have membranes that separate inside materials from the outside materials. We have those because we have a lot of membrane enclosed organelles inside of our cells. This is typical to eukaryotic cells. Different parts of the membranes contain different things. We saw the nucleus, for example, contains all of the DNA, controls all of the stuff relating to protein synthesis. The cytosol has all the other parts floating around in it, and the mitochondria are also floating around in there. Now, most of these reactions that we're going to talk about, glycolysis, for example, that happens in the cytosol. Some more specialized reactions, like the oxidative phosphorylation, will happen in the mitochondria. And so you can kind of see here those designations, cytosol being just the environment of our cells, but inside the mitochondria things get more exciting. The mitochondria has many little folds in its structure called the matrix. And there are essentially there's an inside and an outside of this intermembrane matrix. The inner membrane and the outer membrane have an intermembrane space. So the outer membrane just kind of encloses it. The inner membrane is what's all folded up. Okay, and this intermembrane space, this space in the middle, you're going to see the mitochondria use that to build up a proton gradient. But we'll get there. All right, so just some of the cell components and some of their functions. Again, the ones important for what we're going to be looking at are the mitochondria, and to some extent, any of the digestive, like lysosomes, digestive organelles. Let's talk a little bit about the players, the key players. We're going to talk a lot about ATP. ATP is the energy currency of the cell. Now, ATP is an adenosine nucleotide attached to a ribose sugar, and we've got phosphates on it. And we looked at this in the last chapter, I believe. So the nucleotide plus two more phosphates. ATP is important because we're going to hydrolyze it to do reactions that normally cost energy, and then we're going to form it from ADP when we get to some of our reactions that give off energy. So this is how we store that energy that's created during the breakdown of the sugars and other molecules. So ATP. Now ATP, when we hydrolyze ATP to ADP, so just releasing one of those phosphates generates a good amount of energy. So if we talk about taking ATP all the way down to AMP, so releasing two phosphates, right, we just double that. And essentially, a lot of the reactions in metabolism and a lot of the reactions that we'll look at in glycolysis are what we say exergonic or exergonic or exothermic. They give off energy, or they have a negative change in free energy, in the Gibbs free energy. This means that they're spontaneous and that they are good for us. They give off energy. Now that's coupled with some other reactions that are endogenous, or what we would say endothermic. They actually take energy. And so what glycolysis does is it couples those energy requiring reactions with a bunch of energy giving off reactions so that we can benefit. And so you can see here the step of hydrolyzing ATP that releases energy overcompensates for the cost of what it costs to put a phosphate on a glucose molecule. Because before we get started, before we start to break down glucose, we have to turn glucose into glucose 6-phosphate. Okay, glucose has to become glucose 6-phosphate, and that means it needs to get a phosphate group. That costs energy, but hydrolyzing ATP pays for it. So when we say that ATP is the energy currency of the cell, this is why. Because we can use ATP whenever we want to pay for reactions that would normally not happen. And so we're going to see why the amount of ATP that you get is very important. So self-quiz, go ahead, pause the video, check that out, give yourself a shot. Moving on. Now there are some important coenzymes. Now coenzymes are going to be the things that help move our molecules from, let's say, glycolysis to citric acid cycle. Coenzyme A, for example, is important for that. We're going to look at NADH and FAD, or FADH and NADH and FADH too. Two other coenzymes that are important in the electron transfer. And so the first off is probably going to be coenzyme A. And you can see it here, but I think we have another slide also. So this one's kind of built off of a fatty acid portion of vitamin B. It's got a phosphorylated ADP on it, and an amino ethane thiol. And that's this guy here, amino ethane thiol. So this makes up the coenzyme A portion, abbreviated as CoA. So a lot of these metabolic reactions involve oxidation and reduction. Now, for glycolysis, we're only going to see one of these such reactions, but the citric acid cycle contains even more. And these are electron transfers. And what we're wanting to do with these electrons ultimately is take them to the mitochondria and let the mitochondria make ATP out of them. And that's called oxidative phosphorylation because it requires the presence of oxygen. Oxygen ultimately will take these electrons as the ultimate electron acceptor. And when we give these electrons collected during these reactions to oxygen, we get a lot of ATP out of it. So we need to see how those electrons get carried from these reactions to those ATP making sites in the mitochondria. So this is just a recap of oxidation and reduction. You might remember this from lecture. Let's look at the first. So NAD+. This is a good oxidizing agent. This gets reduced itself, right? That's what oxidizing agents do. It gets reduced from NAD+ to NADH. And alcohol dehydrogenase is the enzyme that catalyzes this reaction. And it would reduce things like ethanol into ethanol, right? Or, as we'll see later, in yeast, it'll reduce acetaldehyde into ethanol. Alcohol dehydrogenase. Anyways, that's NAD+. Now you can see the full structures here. NAD+ in its oxidized form, and then, right, there's an H sticking off of here, and then there's going to be two H's. So it gets reduced. This is how it'll carry some electrons to the electron transport chain. In a subtle variation of NAD+, there's also NADP. And NADP, the difference is that it has a phosphate group on it, where NAD+ normally does not have a phosphate group. So that's the only difference. NADP is also an electron carrier. It can carry an electron or carry a hydrogen atom and some electrons in the form of NADPH. And you can see that one here. The last of the coenzymes that are electron carriers is FAD. And FAD will carry 2 electrons to FAD and become FADH2. And so again, this one is an oxidizing agent that will get reduced. It has an ADP and a riboflavin structure. So you can see that here. So it's oxidized form, no hydrogens, reduced form, hydrogens. And again, the purpose of this is to oxidize other molecules. And so we can see here, this one had some hydrogens on it, and now it doesn't. Losing hydrogens is oxidation. And so this molecule got reduced. And again, the purpose of these guys is going to be to take electrons to the mitochondria. And here we have coenzyme A. So coenzyme A comes from phosphorylated ADP and that pantothenic acid and that amino ethane thiol. And this is the reactive part, this thiol group. This is what's actually going to react with our pyruvate molecule to go into citric acid cycle. So coenzyme A is going to have a role there. And we haven't looked at pyruvate yet, but we will. And there's a reaction actually that'll take the pyruvate and connect it to this coenzyme A. And then we'll produce something called acetyl-CoA. So this is what happens after our pyruvate is going to get reacted. I'm kind of skipping a step, but we're going to look at all the reactions. Okay, but a lot of its job is basically to carry around these acetyl groups, these two carbon structures, and ultimately this is what we're going to see. We're going to see a 6-carbon sugar get broken down into 3-carbon sugars. We're going to have 2 of them. We're going to see those 3-carbon sugars get broken down into 2-carbon sugars, and we're going to lose CO2 molecules in that process. And then we're going to see those 2-carbon sugars essentially breathed out. Times 2. This is also times 2. Yeah, lots of CO2 is going to get breathed out, but that's how you lose sugars. All of these are oxidation steps. And to get from these 2-carbon sugars into the citric acid cycle, they're transferred by the CoA. So pause the video and give that a shot. All right, so let's take a look at the digestion. So digestion of carbohydrates starts in the mouth with the presence of amylase. our enzyme that starts to break down your sugars, and that can be called salivary amylase because it starts in your saliva. Polysaccharides get broken down into dextrins, which are just smaller polysaccharides. Ultimately, maltose, the disaccharide, and then we do get some glucose in the mouth. Depends on how big your starting chains are. Now this stops in the stomach because the pH is too low. Once it gets back into the small intestines, the enzyme kicks up again. There's enzymes that are produced to break down those dextrins into smaller chunks, monosaccharides. We also get lactose and sucrose hydrolyzed, and then the monosaccharides are absorbed in the bloodstream through your intestinal wall. Hexoses like fructose and galactose get converted into glucose in the liver so that glucose can be the main source for our reactions. And you can kind of see that summarized here. So let's look at glycolysis. Glycolysis is the first breakdown, essentially, of glucose into some 3-carbon chunks called pyruvate. It happens in 2 stages, one that costs energy, one that releases energy. We can think of it as a prep phase and a payoff phase. So this happens in the cytosol. I've said all of this. Reactions 1 through 5 are the prep phase. The purpose is essentially, there's a few purposes here. Glucose gets phosphorylated into glucose 6-phosphate. This has multiple roles. One is to go through glycolysis. The other is to go and to make glycogen. Because as glucose 6-phosphate, it can go and form big long chains as glycogen. So there's part of the reason for that conversion. Now, of course, glucose 6-phosphate gets converted into fructose 6-phosphate, which changes the ring structure, right, of the sugar from that to that. It frees up another carbon that wasn't available before. Now there can be 2 phosphate groups on the sugar instead of just 1 phosphate group. So fructose 6-phosphate conversion happens. Of course, the ATP step, I kind of skipped it. ATP was required to add this little purple ball on here, right? And then we're going to add another purple ball. So there was 1 purple ball, now there's 2 purple balls. So those are the 2 ATPs that go into this. Once we get fructose 1,6-bisphosphate, this molecule has committed to entering glycolysis. And it gets cleaved in half by something called aldose, or an aldolase, that makes these 2 molecules here. Now, glyceraldehyde 3-phosphate's the one that continues. Dihydroxyacetone phosphate gets isomerized into glyceraldehyde 3-phosphate. And then we'll have 2 molecules of glyceraldehyde 3-phosphate moving forward. In the second half of the reaction, we get the energy creating phase. So our 2 molecules of glyceraldehyde 3-phosphate get converted into 1,3-bisphosphoglycerate. This involves the reduction of NAD plus to NADH, right? And of course, times 2 on these. Of course, oh, that was right here. That happens first, and then a phosphate gets transferred, creating this really high energy molecule, high enough energy that it can pay for the cost of turning an ADP into an ATP. This is called substrate level phosphorylation. This is making ATP right there in the molecules. Substrates like these guys, substrate level phosphorylation. When it happens in the mitochondria, it's called oxidative phosphorylation. So 1,3-bisphosphoglycerate gets activated, it hydrolyzes a phosphate bond, it makes an ATP, and we have 3-phosphoglycerate. Now 3-phosphoglycerate gets isomerized to 2-phosphoglycerate. 2-phosphoglycerate gets dehydrated into phosphoenol pyruvate, another high energy molecule. Phosphoenol pyruvate pays again the cost of turning an ADP into an ATP, of course, twice, right? And then that leaves pyruvate. And so in those first 2 the first 2 ATPs that we got back, we paid the cost of the prepping phase. So now we broke even. This second step generates energy. And so if this was all you could do, this is at least enough to keep your body warm. This isn't, I mean, and also you can make 2 ATPs from it. Every sugar molecule can keep your body warm and give you 2 ATPs. That's pretty good. But if you're not making ATP, then there's no point in glycolysis. Glycolysis' only function is to make ATP. And again, that's if this is all you could do. We could do more, and so you'll see the process continues. But if this is all you could do, if you're a yeast or if you're a bacteria, then actually you have a problem. You have all this NADH that you made, and you need NAD plus to keep this process going. So we're going to need to figure out something to do with those NADHs to get our NAD plus back. Since we're not going to be doing electron carrying, right, if we're a yeast or if we're a bacteria, because they are anaerobic, not aerobic. They don't use oxygen. So we'll come back to that. But anyways, generating ATP. Just to give you an idea of how much heat this gives off, you can stay warm on the coldest day of winter drinking an ice-cold soda, as long as that soda is not a diet, because of all the sugar in that soda. That soda is going to generate lots of warmth for you. So here's a real quick look at the reactions. So here's reaction 1. Hexokinase puts a phosphate. Kinases, remember, add phosphate groups. So it puts a phosphate onto that sugar. Then we get that isomerization happening, right? Pyrose turning into a furanose sugar. Then we get, or other way around with the sugars, 6-carbon into a 5-carbon sugar. Here we get that additional phosphate group added on. Here aldolase comes in and cleaves this molecule in half. Now, of course, this is probably in its open chain form when it gets cleaved. And we get these two guys. Dihydroxyacetone has to get converted into this glyceraldehyde 3-phosphate. And I think we see that here. A triose phosphate isomerase does it. Triose, of course, because these are 3-carbon sugars now. So now we have 2 of these glyceraldehyde 3-phosphates. 2 steps happen here. There's an oxidation reaction, I should say an oxidation and reduction. We get the reduction here. The oxidation is glyceraldehyde 3-phosphate actually goes into 3-phosphoglycerate. 3-phosphoglycerate. Which you'll see is the reaction product of reaction 7. The point of not just making this and going right on to product A or reaction 8 is that by going into making this molecule, because after the first step is reducing this, and the second step is turning 3-phosphoglycerate into 1,3-bisphosphoglycerate, is that we've created a real high energy molecule here, one that can make some ATP. And so we do. We make ATP. So ATP gets made with 1,3-bisphosphoglycerate into 3-phosphoglycerate. So again, we could have just taken this molecule and ran with it, but by making this out of it and not costing ATP to do it, we were able to make some ATP. So now we have 3-phosphoglycerate, and this was so phosphoglycerate kinase, again, kinases transfer these phosphate groups, took care of making that ATP. Reaction 8 is now taking the 3-phosphoglycerate and switching that around into 2-phosphoglycerate. So we just moved some groups here. Isomerized it essentially. We're trying to get this O phosphate group closer to a carbon that's double bonded, because if you have that, it's going to make that phosphate come off much easier, which is going to facilitate the transfer to the ATP. So now we have 2-phosphoglycerate catalyzed by phosphoglycerate mutase. Reaction 9 forms that double bond on the carbon that we needed. 2-phosphoglycerate and enolase will actually dehydrate this molecule, will lose a water molecule from here and form a double bond. Now this carbon double bond is right next to this ester or this phosphate group, and this will come off very, very easily, and that'll happen in the next reaction. And so now phosphoenolpyruvate will lose those phosphate groups to form ATP molecules again and pyruvate. So these were those reactions in a little more detail. So overall, glycolysis produces 2 NADHs and 2 ATPs for every glucose molecule. We also get 2 pyruvates and some water molecules. Now the pyruvates could go on to make more energy in the citric acid cycle. ultimately on their way to being breathed out as CO2. But if we were anaerobic organisms, then we're kind of stuck here. And in fact, even in our muscles, we run out of air. So pyruvate needs to get turned into something else in our muscles when there's no air around, just like yeast will have to do something with it as well. So we can talk about fructose and galactose. We mentioned that they get turned into to glucose so that they can enter the citric acid cycle and into glycolysis. Kidneys and muscles turn fructose into fructose 6-phosphate. Of course, we spent a couple reactions making that out of glucose in the first place. Galactose reacts with ATP to yield galactose 1-phosphate, which can convert into glucose 6-phosphate. So it can enter when glucose 6-phosphate enters. And you can kind of see those entry points there. Fructose 1-phosphate can also be converted into glyceraldehyde 3-phosphate. Regulation, this is how we turn off these enzymes. Now hexokinase, that first reaction, if there's ATP around, this usually will inhibit that first reaction or that first enzyme. It's also inhibited by high levels of glucose 6-phosphate. If that's around, then there's no reason for hexokinase to be making more glucose 6-phosphate. Phosphofructokinase is also really regulated. This one makes the 1,2-bisphosphoglycerate. This one is inhibited by high levels of ATP and activated by high levels of ADP and AMP. Both of these mean the same thing, right? If you have lots of ADP and AMP, that means you're running out of energy. Pyruvate is also an inhibitor or an inactivator for some of these. High levels of pyruvate will actually turn off phosphofructokinase as well. And then the last reaction, pyruvate kinase, which turns phosphoenolpyruvate into pyruvate, is also inhibited by ATP or acetyl-CoA, which is another enzyme that's important, or another molecule that's important in these reactions. But what I found most interesting too is the amino acid alanine is also an inhibitor. Alanine comes from pyruvate. It's the amino form of pyruvate. So if you have lots of alanine around, it's probably because you have lots of pyruvate around. And high levels of pyruvate mean that pyruvate kinase has been working real good. So alanine turns pyruvate kinase off. It's pretty neat. The pentose phosphate pathway. Sometimes sugar isn't broken down. Instead, glucose goes through another pathway. And the purpose for this pathway is to create some of the other molecules we need. For example, the nucleic acids are made out of the ribose sugars. Well, this is where those riboses come from. They come from the glucoses. Cholesterol and fatty acids also can be synthesized from glucose. Pyruvate can also enter other pathways to extract energy. We talked about the citric acid cycle, and so we can talk about that, I think, right now. No, we'll talk about that in the next chapter. When that's not available, then you have to reduce pyruvate. If you can't oxidize it, you got to reduce it. And so let's look at those reactions. So we'll look at the aerobic one first for comparison. So when pyruvate has oxygen available down the road to accept electrons, then this reaction, this pathway is open to it. So the first step that has to happen is we need to take pyruvate and turn it into a 2-carbon molecule, an acetyl group. And so that requires another molecule of NAD, another coenzyme, this one was Coenzyme A, and the pyruvate. Now they're showing the thiol group because that's the reaction portion. So this pyruvate dehydrogenase essentially releases a CO2, catalyzes the oxidation and reduction. So NAD plus gets reduced, and the pyruvate gets oxidized. And then we get that reaction between the acetyl group that's left over from the sugar and the CoA. So we get acetyl-CoA. Acetyl-CoA will move on to more reactions. When there is no oxygen available, and so no pathway for that acetyl-CoA, then we get the anaerobic conditions. And in this case, our bodies and other organisms can either reduce pyruvate into lactate or into ethanol. For lactate, there's the enzyme lactate dehydrogenase, And the reason that this is important and has to happen even in our bodies is because we need NAD plus for this glycolysis to happen. We don't make NAD plus and NADH as needed. We have a set amount of them sort of floating around. And so if we suddenly turn all of the NAD plus into NADH, then there's no NAD plus. And so something needs to regenerate that. And so we are willing to turn pyruvate into lactate, even if just for a little while. Lactate can go back into the liver and be turned back into pyruvate, and pyruvate can go back and be turned into glucose. We're not worried about, the extra work. We need to get NAD plus back into circulation. And so in your muscles, without oxygen, this is the pathway that happens. And its purpose, again, is to oxidize NAD plus back into something usable. Now, in our bodies, we do something with the lactose. Organisms like that live in sour milk, for example, just make more lactate or lactic acid. And bacteria, I'm sorry, not bacteria, yeast will actually make ethanol. Let's see. The accumulation of lactate causes muscles to tire and become sore. I think we talked about this a little bit. A recent paper that I read that said actually the lactate and the sore muscles is actually helping you to keep going, whereas it's not actually stopping you and hurting you. It's actually pushing you. Mice that didn't have the enzyme to make the lactic acid tired out even quicker than mice that did have it. So we're learning more about the role in that lactate in our muscles. Again, this is just pointing out the fact that if you are an organism that doesn't breathe oxygen, this is the only step where you make ATP, essentially, when you make lactate. Now, you have to go all the way to lactate because remember that regenerates the NAD. That can go back over here and start doing this again. So we make lactate when we don't have oxygen. When we do have oxygen, we'll go this way and continue on to the citric acid cycle. And over here is what yeasts do. Yeast will turn pyruvate into acetaldehyde by losing a carbon dioxide, and then alcohol dehydrogenase will reduce the acetaldehyde into ethanol. Those CO2 that are released initially by pyruvate decarboxylase, this is what adds, fermentation, or not fermentation, sorry. This is what adds carbonation to beer and to sparkling wine and that kind of stuff. All right, and again, the yeast are doing this. The yeast are making ethanol to get back that NAD+. That's the whole point. And we all love that they can do that. Very quickly, we can talk about the fact that glycogen needs to be made, right? If you have excess glucose and you have a lot of glucose 6-phosphate, you're going to make glycogen. So glycogen is the storage mechanism. And this is glycogen here, shown here. It's in blue. Glycogen is synthesized making those alpha-1,4 and alpha-1,6 linkages. High levels of glucose 6-phosphate trigger this. And when you actually have lots of glycogen, glucose gets converted into triacylglycerides and to body fat. So you don't store and make glycogen continuously. Glycogenesis is when you make glucose from non-glucose molecules. Sorry, that's gluconeogenesis. I misspoke. Glycogenesis is when you make glycogen from glucose. So it's called glycogenesis. And you can see those reactions here just involve activated glucose or glucose 6-phosphate basically being linked together with other glucose 6-phosphates. First step though is you have to move the phosphate group to the proper carbon, right? The linkages happen between the number 1 carbon, and so we want the phosphate group on there. So phosphoglucomutase does that step. And then once that has happened, it looks like pyrophosphorylase attaches a uridine triphosphate residue, a UDP glucose, and then once we've got UDP glucose, those can be brought in and added to chains. So that's how glycogen gets synthesized. It's regulated by insulin, activated in the presence of insulin, so a hormone regulator, and glucagon actually inhibits the process. If you need sugar, you're not going to be storing your sugar, so you want to stop that process. Glycogenolysis is the reverse, essentially. This is when you want to break down glycogen for glucose, so you want to get back your glucose. Phosphorylysis and hydrolysis. See if those are shown. So essentially, one glucose at a time gets removed and gets activated to glucose 1-phosphate. And then glucose 1-phosphate gets reacted or turned into glucose 6-phosphate so that it's ready to use. I think I skipped a reaction here. What was reaction 2? Oh, reactions 1 and 2 involve the removal of the phosphate, or the addition of the phosphate and the cleavage of the hydrolysis of that bond, and then turned into a form usable by glycolysis. Reaction 4 dephosphorylation, so glucose 6-phosphatase takes off the phosphate group to put free glucose back into the bloodstream, so it can travel to parts of the body that need it. All right, pause video, give that a shot. And the answers. Here's what I was talking about earlier. Gluconeogenesis. This is making glucose when you don't have any sugar source for glucose. So the liver and the kidneys can actually make some glucose. If our stores get depleted, gluconeogenesis is initiated, and it's got basically the reactions in reverse. We can still get it in a second. Glycogen runs out. We can only store about a day's worth. So when we're out of glycogen, we start taking basically non-carbohydrate sources. We make pyruvate out of them, and then pyruvate enters into the gluconeogenesis pathway, which you can see is essentially the backwards direction of glycolysis. So pyruvate will get turned into oxaloacetate, which is a citric acid cycle intermediate. Oxaloacetate will get turned into phosphoenol pyruvate, which then just gets us back and running again. A couple of those key enzymes: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, putting the phosphate back on, and so on. So that's gluconeogenesis. So you can see this here. So from glucose, we can either go ribose or the pentose pathway to make some of those other molecules we need, DNA, nucleotides, cholesterol, fatty acids, and so on. We can go glycogen synthesis, glycogenesis, or break it down, glycogenolysis. We can do glycolysis, break it down to pyruvate, or gluconeogenesis, pyruvate back to glucose. And if we have pyruvate, we can go into the citric acid cycle through acetyl-CoA. We can make ethanol if we were yeast, and we do make lactate. And we can take lactate and go back to pyruvate, but we didn't go over the details of the Cori cycle. So pause this, give it a shot. And then I believe that's the end of this chapter. So in the next chapter, we are going to take a look at the citric acid cycle and hopefully oxidative phosphorylation. And if we have a chance, we'll look at how much more energy we get from proteins and fats.